Okay, hello there. Hi everyone. I hope you're doing fine. So, I'm ELISA Jean Pierre, and in this presentation, I'm gonna walk you through some basic steps of immunology techniques. I'm a student at University in the biomedical laboratory sciences, and I'm doing this as part of applied immunology assignment. So I'll be answering 4 questions in this video presentation. So let's go through our questions now. The first question I'll be answering is, what are the applications of ELISA in clinical practice? And the second question, how does herd immunity contribute to disease prevention? And the third question is, what are the main types of vaccines and their characteristics? And the fourth question, which is the last one, will be, what is the difference between active and passive immunization? So let's go through our content of the day. Going on the first question, what are the applications of ELISA in clinical practice? So I start by defining ELISA. What is ELISA first of all? ELISA, or enzyme-linked immunoassay, is a laboratory technique which is used to detect and quantify specific proteins like enzymes, antibodies, or hormones, or antigen within a sample, like blood or serum. So why do we need ELISA, as a question you could be asking yourself? ELISA is very crucial due to its high sensitivity and specificity. And before we see the applications, its application in clinical practice, let's see how does ELISA really work. The ELISA process begins with adding a sample in a well which is coated with either antigen or antibody, and then we add a sample which then we incubate at some temperature like +7, so that after the antibody have reacted with antigen, then the enzyme which is conjugated to antibody will be activated. Then the color change will appear, and that color change can be measured by ELISA machine, and its intensity will tell us how levels of those antigen or antibody are in the blood or in the serum or even other sample. So applications of ELISA in clinical practice. Its first application is used to diagnose infectious diseases. ELISA is utilized in diagnosing infectious diseases by detecting specific antigen or antibodies associated with pathogens such as HIV, COVID-19, hepatitis, and Lyme disease. This helps in identification and management of these infections. The second application is the cancer or the tumor marker detection. ELISA can play this important role in identifying the tumor markers such as CA125 for ovarian cancer or for the cervical cancer. This aids doctors in identifying the early cancers and in treatment decisions. Its third application is in the hormone testing. The enzyme-linked immunoassay is widely used for measuring hormone levels, including insulin, for pregnancy detection, or for thyroid hormone like thyroid stimulating hormone. So, and the fourth application which I'll be talking about today is allergy and autoimmune testing. This ELISA technique is effective in determining and identifying the IgE antibodies related to some allergens or some autoimmune diseases. So let us go to the second question, which is, what are the main types of vaccines and their characteristics? So I'll be talking about the basic ones which are common, and first let's understand vaccine first. as defining a vaccine is a biological preparation that provides active acquired immunity to a particular disease by stimulating a body to produce its own immune response. The vaccine is composed of inactivated or attenuated microbes or antigens, which will instruct the body to fight against those weakened or inactivated antigen and provide antibodies against them, which will keep the memory and help to fight when actual pathogen attacks. Mechanism of action, as I said, they make body to produce its own immune response against the attacked pathogen. The first type of vaccine I'll be talking about is the live attenuated vaccines or weakened vaccines. These live attenuated vaccines or weakened vaccines, they have to weaken the germs before they prepare the vaccine, and then that vaccine will be ingested in human, which will then induce immune response against that specific pathogen. Example of live attenuated vaccine include like measles or mumps or rubella vaccines or yellow fever vaccines. And these vaccines, they are criticized for their because they are not suitable for immunocompromised people. They can either end up causing the disease they were supposed to treat. So the second type we'll be talking about is the inactivated vaccines. These inactivated, as you can hear it, we are talking about killing the germs and removing some antigens, but the pathogens or microbes are literally killed in this type of vaccines. For example, we can have the polio or hepatitis vaccines. They are classified under these inactivated vaccines. The third type is the subunit or recombinant vaccines. The subunit is the vaccines which contain some parts or small parts of the pathogen. For example, it can contain like a protein or a specific carbohydrate from the actual pathogen, but doesn't contain the whole pathogen, inactivated or attenuated. So these vaccines only contain certain pieces of germ, such as protein or sugars, rather than the whole pathogen. And example, we can have also hepatitis B and human papillomavirus vaccine. These vaccines are better because they do not induce whole pathogen into the body. That's why they are preferred most of the times. The next we have the toxoid vaccines. These toxoid vaccines will contain the toxin from the bacteria or the virus we are attempting to treat. Example, we can have like tetanus or diphtheria, which falls under this type of vaccines. Now going to the next, we have like the messenger RNA vaccines. The messenger RNA vaccines, they contain messenger RNA, and messenger RNA, as you know, contains genetic information which will instruct the cells of the body to produce that specific antigen, and then the body or the immune system of the body will produce antibodies against that specific antigen produced. For example, we can have under this messenger RNA vaccines, we can have like the COVID-19 vaccines like Pfizer or whatever the many vaccines are given falls under this part of messenger RNA vaccine. The next question, the third question, is how does herd immunity contribute to disease prevention? We start also by understanding what is herd immunity. The herd immunity is like when the bigger vaccinated population prevent older unvaccinated people from that specific disease. Here, some people are vaccinated, and so they cannot transmit the disease to those who are unvaccinated. And this time of vaccines is mostly traditional somehow. And you can see the control, the importance of herd immunity. The herd immunity is vital for controlling outbreaks of contagious diseases like measles or polio. Like in this way, if the disease outbreak in a specific part and they are vaccinated, they cannot spread disease to the neighboring places or regions. The second role is protection for vulnerable population. Some population or some people are not suitable for vaccines because maybe they are immunocompromised or because they are allergic to that specific vaccine. So by using this herd immunity and vaccinating some of the members, they can prevent those who are not vaccinated. And the third is the reduction of healthcare burden. By lowering disease incidence, herd immunity reduces hospital visit and healthcare costs associated with treating infectious diseases. So we don't have to vaccinate all people, but if the larger population is vaccinated, it can indirectly prevent those who are unvaccinated. And going for example of herd immunity, an illustrative case is where 90% of a community is vaccinated and 10% is not vaccinated. And for example, like in smallpox cases, they can vaccinate some and don't vaccinate others. Going to the last question, what the difference between active and passive immunization? Okay, we'll start by understanding them. And first, going with immunization. What is immunization? Active immunization involves stimulating the immune system to produce its antibodies against a specific pathogen, usually via vaccination. This is acquired immunity, is long-lasting, and it can sometimes last for whole life because the body is stimulated to produce its own immune response and keep memory of it so that in future recurrences it can fight it easily. The passive immunization. In this kind of immunization, the patient receives the antibodies or receives the serum which contains antibodies from another person who have recently recovered from the disease or who had been vaccinated. Going through the differences on how they work. Active immunization offers a long-lasting immunity as the immune system learns to respond to pathogen here and respond actively to that pathogen and keep a memory of it. And for here, the active immunization is typically in vaccines which will stimulate the body to produce its own immune response. Application in healthcare. Situation for active immunization. We have active immunization. They are recommended for long time and for strong immune response in future recurrences. While for passive immunization, it can be used when the patient is in emergency case or has affected with specific diseases which cannot allow body to make antibodies directly. So while the body is waiting to make those antibodies to fight against that specific pathogen, the patient may receive some antibodies from another one or from another individual, which will be helping him or her in that situation. So thank you all for following. This is my reference which I have used, and this is the end of my video presentation. Bye bye.